Hi, I'm Robert Smith. The second edition of my ebook, Medicinal Chemistry, A Fusion of Traditional and Western Medicine, has been published. It can be used as a textbook for the first semester in a two-semester course in medicinal chemistry. The third edition can be used for the second semester, while the first edition is for a one-semester course. The first and second editions begin by describing a newly emerging way of maintaining health and fighting diseases. It is based on a fusion of systems thinking that is typical of traditional medicine and reductionist thinking that has been typical of Western medicine. This new way of thinking, doing research and communicating, is affecting the way new drugs and natural substances are being developed. It is also affecting the type of education that pharmacists, physicians, medicinal chemists, and even lay people are receiving. The second edition goes into much detail about recent advances in our understanding of genes and different types of DNA. So instead of emphasizing how genes code for messenger RNA, which codes for proteins, we learned that there is not a strict hierarchy of DNA causing RNA to be made, which causes proteins to be made. There are also mobile genetic elements, epigenetic changes, and post-translational changes in proteins that can affect the phenotype or the composite of an organism's observable traits, including one's health. The second edition also contains a chapter on biotechnology that was not in the first edition. It tells how biological tools are used in many different fields of technology and how technology is used to study biology and develop new drugs. For example, genetic engineering tools are used to solve the human genome. These tools are leading to the production of new types of bacteria, algae, and other life forms that will make many of the products that are now made from fossil fuels. Biotechnology is also used to identify DNA at crime scenes and analyze DNA from extinct species. It has been and continues to be used to identify genes, new therapeutic targets, and to enable important advances in personalized medicine. For example, a recently approved flu vaccine, Flublok, is made by insect cells, and transgenic goats are used to make a recombinant human antithrombin for treating hereditary antithrombin deficiency. Moreover, whole cells from a patient can be used therapeutically. This includes immune cells for leukemia, glial progenitor cells for restoring myelin to the central nervous system, embryonic stem cells for type 1 diabetes, and even fecal transplants for MRSA infections. The second edition also goes into more detail than the first edition and updates information presented in the chapters on how drugs are developed. This is especially true for personalized medicine and the way individual differences in genes coding for metabolic enzymes can affect the potency and toxicity of drugs. One of the advantages of ebooks is they are published rapidly, making it easy to update subsequent editions. For example, the epigenome has become a good target for therapeutic drugs that are being developed, so this is described. My ebooks can also serve as references for people working in the pharmaceutical industry and as a source of information for patients and lay people. They take a multidisciplinary approach since teachers, pharmacists, doctors, and dentists are often asked questions by students, patients, and friends that are not answered by other books with a more limited scope. So, it is important to know not just how new drugs and dietary supplements are developed, but also how they can affect the whole person who may take them. It is also important to know how environmental toxins like bisphenol A or BPA can affect health. The goal is to inform professionals and non-professionals so they can communicate with each other and make the best decisions on how to prevent, treat, and cure diseases. The basic science behind drug development is discussed, including identifying a lead compound and showing the proof of principle behind its effects. The appropriate dose and mode of delivering the drug are also determined. Next, higher doses need to be given to test animals like mice or rats to look for possible toxicities. After doing these studies, it is often necessary to improve the lead compound to make it more bioavailable and less toxic. All this research must be done in compliance with good laboratory practices, or GLP. Next, enough of the drug must be manufactured for clinical studies. It must be made in accordance to the principles of current good manufacturing practices, or CGMP. The basis behind CGMP is analysis, so many different ways of analyzing drugs are discussed. Unlike other books, much detail is given on analytical chemistry and the details of GLP and GMP in this book. My ebook also goes into detail on how new drugs are investigated in clinical trials, from phase 1 testing on healthy subjects to phase 4, in which the safety of approved drugs is monitored. The book continues by talking about preventing diseases through proper nutrition. The differences between prescription fish oil and fish oil that is sold over the counter is mentioned. The difference between omega-3 fats and fatty acids is clarified. Genetically modified organisms, or GMOs, are also described, as are antibiotic-resistant bacteria. I hope that you will find this book to be useful and informative.